Okay, welcome again, everybody, to Biological Signaling 2. We are in the middle of somehow solving the mystery between the crack channel proteins. You know, the crack channel is the calcium release activated calcium channel that is so important in mast cells, actually, to drive the mast cell degranulation by calcium influx. And keep in mind how this crack channel gets activated. The main signal to activate this crack channel in the plasma membrane is the depletion of the ER calcium stores. So calcium needs to somehow get dropped in the lumen of the ER, and this occurs by the second messenger IP3 through an efflux of calcium by IP3 channels into the cytosol. But you remember that it's very important to have this sustained long calcium signal in the cytoplasm to drive the exocytotic process. And here it's particularly important that this can only be gained by a calcium influx through the calcium channels in the plasma membrane. And we just discussed a protein actually in year 2000. It was a Nature paper that people thought from a TRP superfamily, TRPV6, or also CAT1 called, is the protein behind the calcium channel in the plasma membrane. So the main point was, is this true or not? And let me just show you how actually finally it has been proven that this is not the right protein we actually are looking for. So we have two models here. On the one, we have the RBL mast cells that contain the endogenous crack machinery. So they have these endogenous crack channels expressed. And we have the JKU293. These are the HEC, the human embryonic kidney cells. That a very nice expression system to study, for instance, the expression of CAT1, or how it's also called TRPV6. We typically use, because we want to detect, to measure also biophysical characteristics of a current through these channels, we use electrophysiology, we use the patch clamp technique in the whole cell configuration. And as I just mentioned, there is the need to deplete the calcium stores. And there are two possibilities how you can easily do it. The one is, although it's not, let me say, directly as it physiologically occurs, because physiologically, that's summarized under the active store depletion. That occurs at the somehow resting calcium concentration of calcium 60 nanomolar, and in the pipette there is a high concentration of IP3 given. That is the second messenger, and via the pipette it diffuses into the cell, opens the IP3 channel, leads to a drop in the calcium concentration in the lumen of the ER, and in turn you should see quite an active, a rapid activation of these crack currents. However, the passive store depletion works via a chelator of calcium, EGTA, that is also put in the pipette, which diffuses upon establishing the whole cell configuration into the cell, into the cytoplasm. It grabs all the calcium, and you remember we have always a certain leak in the ER membrane. So a certain amount of calcium is always flowing out of the ER, but usually it gets pumped back by calcium ATPase in the membrane of the ER. But what now happens is the calcium which goes through the leak into the cytoplasm binds with a high affinity to EGTA. So there remains no calcium to get pumped back. That means over a certain time frame, and that of course takes longer. That is why it's also called passive store depletion. That usually takes 2 to 2 minutes, something about this. Whereas on contrary, this occurs in the range of about 15 seconds, something like this. But the effect is the same. On the long-term range, under both conditions, the ER stores gets depleted, and you can see a very nice calcium current develop. The classical technique is, of course, the patch clamp technique, but you will also see some experiments using fluorescence microscopy. So here are very typical whole cell recordings of the calcium current. And remember, we have here the two cell system, the RBL systems. They have the endogenous crack current. That was actually a cell which has been used in 1990, 30 years ago, to demonstrate that there is a crack channel, a highly selective calcium channel in those mast cells present. And these are red basophilic leukemia. Remember, basophils are also among those cells actually that drive or that can take part in allergic reactions, because they also contain these granules ready for release, the allergic mediators. So what is done here? We have, just that you understand how actually these plots are obtained, we have here current versus time. And time zero means here the whole cell recording configuration is established. And from that time on now, you have these perfusion, or let me say you have this infusion, the diffusion of the pipette solution, which within about 2 minutes completely substitutes for the ionic solution in the cell. So here we have at that time point starting EGTA infusing into the cytoplasm of these RBL mast cells. And you can nicely see here, it takes some time. At the beginning, this is the calcium current, usually due to conventions depicted as a negative current. You see at the beginning here is no calcium current present. But after some time, when the more EGTA gets into the cell, the more the ER stores gets depleted. You can see here an ongoing increase in the calcium current till it reaches about the maximum in the range of about 150 seconds. Whenever we check here for this calcium current, we do this also by doing current voltage relationships. And meanwhile, you also know what the current voltage relationship looks like. You can see here its potential from -80 to +80 versus current. And you can see in particular, the more negative the cell is in the inside, the higher the current is. However, at the beginning, the current is pretty small. That relates here when it's just actually at the starting point. The current is pretty small. And then over the time, we reach a certain maximum. And then it looks like this. So this is current voltage relationship with a reversal potential in the range of a plus 40 millivolts or plus 30 to 40. So it's positive, and due to many other experiments, control experiments, we know that actually comes or is an indicator for calcium selectivity. So that is the endogenous current. We actually looking now whether our protein of interest, that is the CAT1, can also somehow substitute or is the key protein behind the crack channel in the plasma membrane of RBL mast cells. But you can see here one big difference. Whenever we check for CAT1 currents in these human embryonic kidney cells, the main difference is at the very beginning, we already have these currents highly activated. You can see here, we already start at the maximum, and then maybe it declines a little bit, maybe due to this increased calcium influx. There were processes going on of the cell to somehow prevent an overload of the cell by too much calcium flowing in. But you can see here, okay, that's of course a very different behavior. It's not under the control of the store, because although of the stores, they are pretty full in these human embryonic kidney cells, and maybe you get a little bit of, you get, of course, here the same effect that the more EGTA diffuses into the cell, the more the stores gets depleted, but you can see there is no further current activated, because that's okay, because the hex cells per se, they do not have this crack current. But when now we express CAT1 in the hex cells, you can see, okay, they have a selective calcium current, as you can see it under A. So it also shows this positive reversal potential. And it looks somewhat similar to that current here, where it's fully activated in the mast cells. But the main difference is in these cells, it's not under the control of the ER stores. And actually, we are looking for a store-operated calcium channel, this crack channel, but this seems not true as long as CAT1 is expressed in the human embryonic kidney cells. So it was quite clear. Let me say, meanwhile, from many experiences of other scientists, that the surrounding, the environment, the cell where a protein is expressed could also determine the behavior of this protein. So although it's the same protein, but there is a whole cell machinery behind that actually helps to control this protein. So the question, of course, was maybe CAT1 is only not in the correct environment. What happens when we express CAT1 in these RBL mast cells? And that can be seen here. And that's a very, very different behavior and yeah, a very nice change in a way. You can see here in black, this is passive store depletion by EGTA. This is active store depletion by IP3. You can very nicely see this is the endogenous current. That also occurs, of course, in control cells. But when we now express in addition the CAT1 in the same cells, we have, of course, an overlap of the endogenous currents plus now that current that actually develops due to the expression of CAT1. You can nicely see here a big change. CAT1, when expressed in the RBL mast cells, it's not activated at the very beginning, which is called a constitutive activation. It's always activated. It actually takes some time. It actually takes almost the same amount as the endogenous current before it gets activated. So it seems when CAT1 is expressed in the same environment as the endogenous crack channel, it also behaves. It gets under control of the ER stores, and you can see here it reaches the maximum in about the same time range between 100 and 150 seconds. The same holds true when we do the active stimulation now by IP3. You can see here on the time scale, here activation occurs much faster under both conditions, the endogenous crack current, but also the current that now in addition is obtained through the activation of the CAT1 channels expressed in RBL cells. You can see here in a range of about 50 seconds, you already reach a maximum. And you do see not here a delay because IP3 is diffusing, and at the same moment when it's inside the cell, it binds to the IP3 receptor and starts to deplete the stores. That's the reason why this is pretty fast compared to EGTA, which only prevents the calcium pumping back into the ER stores. So that is only works at the, as I mentioned before, it's a much longer time frame. So very nicely, CAT1, when expressed in the same cell environment, in the same cell, in these RBL cells, it seems to behave in a similar way as the endogenous crack channel. So that could be, and that has been taken in the Nature paper as an indicator, okay, maybe this is indeed the crack channel, the protein behind the crack channel. But there are further biophysical characterization going on, and that's known for a long time, that calcium channels, they can also pass monovalent ions. Let me say sodium, but only when all the divalent ions in the solution, particularly calcium or magnesium, are taken away. They need to be substituted and not put in the solution. And this solution where no divalent, where no calcium and magnesium is present anymore, this is called a divalent-free solution. A divalent-free solution. That is a solution that particularly contains at the extracellular side mainly sodium. And you should get now sodium flowing through the crack channels. That's well known. You can see here that's already, so the time scale, it might be a little bit misleading, but we start here when all the crack channels already have maximally activated. So that's now actually, it should be here about 150 seconds, because we do see already the crack channels have activated, and we make now a switch from a calcium containing solution to a divalent-free solution. There are no calcium and magnesium present. You can see here this increase because even we have a higher, typically with calcium, we can use here in those experiments 2 to 10 millimolar calcium, but here there's a higher concentration of sodium that's about 145 millimolar outside. So you see an increase in the sodium current, and you can see it here as well, a change in the current voltage relationship. But keep in mind, here you see this increase, but you still see, even with sodium, this very positive potential. When you now check for the store-operated CAT1, and of course, this is an overlap which contains the endogenous, but also the CAT1 expressing channels that are now also allow the permeation of sodium in the divalent-free solution. You can see here also an increase. But what it's dramatically different here is some of these U-shaped form is apparent here. You can see when we do here a current voltage relationship, you can see there is a U-shaped form at the beginning, and then maybe it has a different form, and additionally it has also another form, because it seems that here the reversal potential is shifted from a high positive reversal potential to about 0. So one point actually, when we rechecked the Nature paper here, because they made similar experiments, what somehow maybe what was misleading, they somehow cut this current voltage relationship. Then they only presented it only to a range of -60 millivolt. So they did not show this different behavior. But this is a very, for biophysicists, this is a very characteristic hallmark of these CAT1 or later on of TRPV6 channels, that they show this U-shaped form at the very beginning when doing the current voltage relationships. In this Nature paper, they omitted it. They didn't show it. So to make it quite similar to the endogenous current. But that was a very, very clear characteristic. Okay, the expressed CAT1 channel, at least based on biophysical characteristics, is distinct, particularly in the divalent-free solution. The current voltage relationship looks distinct compared to the endogenous crack current. And the next point was, of course, to use inhibitors that have been known to inhibit these crack channels. And let me first show you the action of the inhibitor. That is a compound you do not know, you do not need to remember the name. It's only called 2-APB, but it's a very, very compound that has been developed for several years meanwhile to be a blocker of crack channels. Let me just switch here to the endogenous crack channels you see here in RBL mast cells. So here also the currents are completely activated. And when you apply now 2-APB, you can nicely see here this inhibition of the current. It's not fully inhibited, but it's inhibited by about 80%, something like this. So this is because also that was clear, that was expected. Let's first check what happens when CAT1 is not in the correct environment. That is CAT1 expressed in the hex cells. And you can see, okay, we have here a current at the very beginning because it's open almost at the very beginning when we establish the whole cell configuration. So that's the reason why the current here, the maximum is set to 100%. You can see this very high current. You can see when here 2-APB is applied, the different actually occurs. There is no inhibition of the current because then the current should get less in percentage. But you can see even there is a slight activation of the current by 2-APB, which you can also see here. There are these are of course similar with calcium current voltage relationships compared to the endogenous current. But you can see here in hex cells, CAT1 expressed behaves in terms of what the 2-ABB blocker is doing completely different. You get a slight increase, but of course you will not see an inhibition. However, when CAT1 is expressed in the RBL cells and is now under the control of the stores, so here at the beginning we have already the stores depleted, so we have here endogenous plus CAT1 currents activated, and you can nicely see here when CAT1 is expressed in the RBL cells, 2-ABB works pretty well. So here the same. Whenever it has the same environment as the endogenous cracked channel, okay, 2-ABB works in a similar way as with the endogenous cracked channel. But that is a matter of the environment. So let's summarize. There are, of course, similarities between CAT1 expressed in RBL cells in the correct environment in terms of they are blocked by 2-ABB in a way that they behave similar to cracked channels, and particularly that they are under control of the ER stores, meaning when the stores are depleted, the more of these CAT1 channels get activated. However, based on a biophysical comparison, the monovalent current voltage characteristics of a cracked channel and of CAT1 are distinct. Here we have this, that is called an inward rectification in terms of the endogenous cracked channels, but in terms of CAT1, you see this U-shaped form at the beginning, and that's clearly a distinct feature that is not visible with the endogenous cracked channels. So, of course, maybe the idea came up. It could be because usually all these channels, they work in a way that you have a tetrameric assembly, meaning four monomers have to come together to form a functional channel. And you can see here a cartoon actually of a CAT1 monomer with these classical 6 transmembrane helices. Here we have the P-loop, there is the selectivity filter, which gives the high selectivity for calcium, and some additional structures here. We will talk about them later. That is an ankyrin-like repeat that are actually protein-protein interactions domain, from which it's known that they play a role, particularly because with monomers, they have to find each other due to inherent affinities in the plasma membrane and build up a functional channel with a functional pore. And of course, transmembrane interactions between the transmembrane helices, each one point where an affinity exists, but these ankyrin-like repeats also play a role in driving the assembly of these tetrameric formed channel. And the idea is maybe the current voltage relationship is in as far different because in the endogenous cracked channels, not all of these 4 monomers that come together are identical, but maybe 2, that's just a cartoon, just a hypothesis, but maybe 2 actually are derived from a CAT1 protein, and the other 2 we do not yet know. But it could be that CAT1 somehow contributes to the endogenous cracked channel, but it's of course not a homomeric identical formed tetrameric assembly from 4 identical CAT1 monomers. So that is an idea. Is CAT1 a subunit of crack? So let's first have a look whether actually we can measure the assembly of CAT1 monomers, that it occurs in a way that was just hypothesized. And for this, a very nice technique can be used. You have already heard about that, particularly those who have done the characterization of biological nanostructures in the bachelor. Here, this technique has been introduced, the FRED technique. And it works in a way that somehow to visualize, to get a signal out whether proteins are in close proximity to each other, we have to label the proteins. And the very classical way to label proteins is to use variants of a green fluorescence protein. Actually, that is a protein, and the fantastic way that was also why the Nobel Prize several years ago was given to those 3 scientists who developed these green fluorescence proteins and also other derivatives with different colors. They are proteins because at that time point, all of these fluorescent dyes that have been developed are all small chemical molecules, but they cannot be so easily attached to proteins. Particularly, they could be attached to proteins in solution when they are purified, but they are more difficult to attach to proteins which actually occur in a living cell. The most important point is whenever you have a protein, you have also the DNA behind it. And based on molecular biology tools, you can easily somehow hook CFP or YFP. These are two variants, the cyan and the yellow variant of a fluorescence protein. You can hook these two fluorescence protein at the level of DNA to the DNA of a cut1 channel. And then you get always a cut1 channel where particularly at the amino terminus is in front of that amino terminus is either CFP or YFP. You know, one of the important points to use here the correct pair of these fluorophores is that because there is this very nice first resonance energy transfer possible, meaning that when the CFP gets activated by a certain light, then you can see that the emission, this is the CFP emission that is under that peak, the CFP emission, the light that is emitted from CFP shows an overlap with the excitation, that is the excitation spectrum of, that is the dotted line for YFP. And you can see there is a certain extent of overlap. And that means when these CFP and YFP are closer together in a range of about closer than 10 nanometers, then you have a radiation-less transfer of energy from CFP directly to YFP, meaning when you excite CFP and it's closer than 10 nanometer to YFP, you get direct emission of a YFP fluorescence signal. And this is particularly a fantastic tool to come down to distances, you see, where 50% of energy is transferred. That is in the range of about 4.9 nanometers. So you have a fantastic resolution that would not be possible when you only have one fluorophore, because with these fluorophores, the resolution typically to see whether they come closer together is in a range of about 200 nanometers. Whenever fluorophores of only one type come closer together than 200 nanometers, they would not be resolved anymore by each other. But whenever we have this CFP and YFP, due to this thread, which shows this transfer of energy only at very small distances, this is a distance range where we can extrapolate or hypothesize that this is indeed an indication of protein-protein interaction. So let's use these two fluorophores, CFP and YFP, hooked on to two different monomers of cut1. And let's see here, this is when they are expressed. This is the CFP in a cell. Of course, in the same cell is also, in the same cell is also YFP expressed. What we can do now, we can excite the CFP and look on the YFP emission. So this is the thread. Maybe you of course, as you have learned, you have to do some calculations to correct from some signal overlap here, but then you can see here, the higher the thread, the more brighter actually the colors get. And you can see here, particularly close to the plasma membrane or in the plasma membrane, we have here dots where actually we do see an interaction between YFP and CFP labeled cut1. So we can use thread. We can use thread somehow to detect the interaction. And one point which is pretty clear, these molecules, these cut1 monomers, they do interact with each other. So as I mentioned before, this interaction is in part driven by the transmembrane domain. So here it's shown still for a homomeric cut, but at least when we have a heteroimeric cut where the two other proteins, as we hyperphased before, are yet not known, the interaction between two similar cut monomers also involves transmembrane helices, but also this ankyrin-like repeat which occurs in the amino terminus. These are also important domains for protein-protein interaction. And here, that's a very, let me say, nice tool that can be used here to interfere with this assembly of a functional cut1 derived or cut1 including tetrameric channel. Because we can say, I can only express in cells the amino terminus of cut1 that includes these ankyrin-like repeats. And of course, the ankyrin-like repeats, the amino terminus, the ankyrin containing the ankyrin-like repeats, it will bind to the other amino terminus of a functional, or let me say, of a proposed assembly of cut1 channels. But what is missing is the whole transmembrane portion. So whenever the whole transmembrane portion is missing here, we have only here 3 fully functional cut1 proteins. But what is missing is the 4th, and particularly here, the pore loop is completely missing. And we only have three of those that make a pore. 3 pore loops are not enough to make a functional channel. Here, actually, you need 4 pore loops. So what we can use here, the amino terminus can be used, which is called as a dominant negative species, because it depends, of course, the affinity, but whenever it binds, let me say, to in a way that you have a tetrameric assembly where at one side only the amino terminus is sitting and not the full cut1 channel, this will lead to a non-functional channel. So it will lead to an inhibition of those channels and to a decrease in the current. So let's check whether this very nice tool indeed works. And you can see here, first, of course, it was checked cut1 expressed in the hex cells. This is, as already mentioned before, the cut1 fully active in these cells, not under control of the ER stores. But of course, the mechanism should work independent, whether these cells actually are controlled under the ER stores or not. And you can nicely see it here when cut1 here, in addition, this amino terminus is co-expressed. You get a very clear decrease in the currents because it seems the amino terminus portion of cut1 binds to the cut1 channels, and you have here channels where one transmembrane unit is missing because here sits only the amino terminus with these ankyrin-like repeats binding to the other 3 or one of the other 3 monomeric assemblies. And this leads to a very clear decrease in the current. So this is a sort of a proof that we can use the amino terminus of cut1 to interfere with cut1 derived currents. And you can see there are also, I mentioned before, within the TRP superfamily, there are also other channels which are the TRP, belong to a TRP C3 class. This is called TRP C3. TRP C3, here actually a C is missing. They have a similar structure. They also have the TRP C3 ankyrin-like repeats. But that has been tested here to really show that the amino terminus of cut1, or TRPV6 actually, how it's called, because it belongs to the same superfamily TRP channels, that the amino terminus of cut1 is indeed specific to cut1, because when we overexpress or co-express cut1 channel with the amino terminus against TRPC3, which you can see here, on the levels of domains, it looks pretty similar, but on the levels of interaction, you can see the amino terminus of TRPC3 is not able to inhibit cut1 derived currents. So that was taken as a sort of specificity. What we use here has a certain specificity to cut1 derived channels. So let's have a look. What happens to the endogenous crack channel in a way that we let us presume that maybe the crack channel contains one or two subunits within this tetrameric channel assembly, and we can interfere in this assembly because we use now the amino terminals of CAT1, and that was quite, let me say, a fantastic result at first sight, that the crack channel, when we now express in the RBL mast cells the amino terminals of CAT1, we get then complete inhibition of the endogenous crack channel. So that was at first sight a very nice proof, because we tested for specificity here with these N-terminus of CAT1 versus N-terminus of TRPC3, and at least here it looked quite specific. So this could mean that indeed CAT1 proteins are contributing to the tetrameric assembly of an endogenous crack channel. The N-terminus of CAT1 may directly interact with crack channels or may interfere with the coupling machinery. That's not fully clear. That cannot, but the more direct, let me say, the more direct conclusion would be directly interferes with the establishment of a functional CAT1 channel. So we also tested this in terms of, just to present you another assay, in terms of calcium imaging, because also in these cells you can nicely do calcium imaging. We have heard of this. This is with FURA2, and it allows you, is a ratiometric dye. It allows you at two different wavelengths as a ratiometric signal to detect changes in the intracellular cytoplasmic calcium concentration. You can see here in control cells, here a compound is used, but that's just an additional information. A compound is used that also leads to depletion of the stores. And you can see, and that usually is applied here when the stores gets depleted. And here at that time point, calcium is given. So this increase is an indicator that we have here calcium flowing through the crack channels. And when now we have channels actually, and we can nicely see here, because the amino terminals, that's also marked by a YFP. So you can see the cells where it's actually expressed, and you can nicely see here, when we have now with the endogenous crack channels, the amino terminals of CAT1 expressed, you can nicely see here, there is a clear decrease in the increase, or a clear decrease in the cytoplasmic calcium concentration, which is obtained when calcium is put to the extracellular solution compared to controls. So we have here quite a similar result. The endogenous crack channel leading to this calcium influx, that is inhibited when the cells express here the amino terminus of CAT1. We can also measure directly the degranulation by terms of, because the histamine, which is released from the granules, can also be measured. So you can see that is a longer time frame, at least to get full release. This is in control cells. And actually, when we calculate here on average here, how much the release is reduced when we express in those cells the amino terminals, you can see here there's not that much inhibition of the release of histamine. But we have to take into account, because whenever you transfect cells, the plasmid, meaning these amino terminals of CAT1, is not taken up by all of the cells to 100%. So here actually the cells have been selected in a way that only those cells have been measured here that show a signal that they have taken it up. But here it's not, in this assay, we cannot directly measure the cells that have taken it up, but we have used this. So this means we have about only 40% transfection, meaning only 40% of cells, and these 40% have been taken here. But here we cannot decide whether cells actually have or have not taken up the amino terminals. But when we correct it only for those cells that has taken it up, meaning we have 40% of cells that have these amino terminals transfected. You can see here it looks pretty much the same, and we would expect here a decrease in the histamine release due to these expression of the amino terminals of CAT1 and the blockade of the calcium influx through these proposed CREC channels. So that was a very crucial experiment, but it tells you what you see right now, that you never can do enough controls to really check is what you are here proposing, is this really a correct result, or of course are there other interpretations? And the main question is, how specific is the amino terminals, the tool we used, the amino terminals of TRPV6, in terms of an inhibition of a TRPV6 CAT1, or let me say in terms of CAT1 channels? How specific is this really? So another tool, which is meanwhile, maybe it's still used, but I will come to that in a second, but we have meanwhile additional techniques at hand, is to use siRNA, or it's also called RNAi. That are small, and there the name comes from, small interfering RNAs that are directed against certain proteins. And that is a mechanism which is also used physiologically by cells to somehow make RNAs for a whole protein unstable, so that this RNA is actually degraded and you will never end up with a real protein. So this can be somehow seen as a knockdown of specific proteins in living cells, because the siRNA is specific. They can be specifically constructed against CAT1. And first of all, when you can order this, and there are also programs how to design RNAs. And when we have got these RNAs specific against CAT1, of course, first of all, you have to prove it, and best in a different system. We know that LN cap cells that are prostate cancer cells. And we know that these cells, that was shown by other scientists at those times, that those cells, they have, first of all, expressed TRPV6 or CAT1 proteins, and second, they have a very nice, very similar looking CREC current. You can see here the CREC current. It takes some time to get activated. It seems also to be controlled in these cells under VER stores, but you see the activation within 100 seconds, they get activated, and you can see here the current voltage relationship looks pretty much the same as the endogenous CREC current in mast cells. But that was shown for clear, this is and occurs because here, this is mediated by CAT1 proteins or TRPV6. So when we use now siRNA and treat the cells with siRNA against CAT1, here that was expected. We should, of course, see a decrease, an inhibition, because the protein now is expressed to a less extent, because siRNA against CAT1 destabilizes CAT1 in these cells. Usually you have to treat the cells for about 3 days, or wait for 3 days. And then you have the turnover, because you have a certain protein in the plasma membrane where it stays for some time. But 3 days, it's enough when you prevent protein synthesis, you prevent synthesis of new CAT1 proteins for 3 days. You can really be on the safe side that the other in an endocytotic process has been taken up, which was previously in the plasma membrane, and no new now CAT1 protein is synthesized because of the siRNA. So this leads to this inhibitory effect. Let me just say, meanwhile, one technique is very popular. This is the CRISPR-Cas9, and maybe all of you have already heard. That was also the Nobel Prize, I guess, last year, if I remember correctly, or 2 years, but I think last year. it's also another technique. It's quite more complicated, although it sounds very, very easy to be done, where you also can interfere at the level of DNA and prevent expression of certain proteins. The main difference here, after about several days, at day 3, it's pretty fine. But when you wait on for about 1 week, the siRNA is already destabilized and has left the cell, and CAT1 would be expressed again. With this CRISPR-Cas9, you get actually a change, a disruption of gene expression or protein expression at the level of the nucleus in the DNA. And so this is a permanent disruption. So that is easier to use, but it's not easy to get. You need a lot of experiments. We tried it months in my group, and although it looked quite promising, finally it turned out, okay, these cells still, they express CAT1, or at that time we used it for ORI channels. So you have to do also a lot of controls, because also with CRISPR-Cas9, you can get off-target effects, and that also needs a lot of controls. But let's keep to now the siRNA application against CAT1 in the RBL mast cells. And that was one of the crucialest experiments in this whole study. And you can see here, black is the endogenous current. When now these cells are treated against CAT1, with this specific siRNA, and we can say this RNA, of course, it's much more specific than the amino terminus of TRPV6, which has been used before. But you can see here, there is no change, really no change in the crack current activating, meaning it can't be that CAT1 or TRPV6 contributes even in terms of 1 monomer, 2 monomers in a tetrameric assembly, because then when we somehow knock out CAT1, there should be a clear decrease in the crack current, but that is not seen here. So from these experiments, one can very clearly conclude CAT1 is not part of the endogenous crack channels of endogenous crack channel in RBL cells. And just to be sure that the siRNA are working, because we have 2 different cells, it worked pretty well in the Allen cap cells. But the question is, okay, maybe something is different with the RBL cells. We did the same experiment in RBL cells. We expressed here at the same time, of course, overexpression of CAT1 leads to higher currents. That is what I've shown before. And when we combine here now CAT1 at the same time put into the cell together with siRNA, as soon as CAT1 enters and is put in the machinery to make a protein, the siRNA is present at the same time, and it destabilizes and prevents expression of the CAT1. And that actually is seen. You see a clear decrease here to a signal that is left because of the endogenous crack channels, which are not subject to a reduction by treatment with siRNA. So keep in mind, the better, the more specific your tool is you have. And of course, the better, the more controls you have to come up to a conclusion, the higher is your chance that you actually publish something or report something or put something in your master thesis or even in your doctoral thesis that presents a correct result. Finally, from these experiments, we conclude CAT1 protein is not a subunit of a crack channel. So that was quite an important message, because that was also later on confirmed by other scientists as well. It makes very clear that the Nature paper, their message of TRPV6 as a protein of a crack channel actually was wrong. A 2ABP sensitive component regulates activation of both crack channels and CAT1. That's clear. There is a similarity, because 2ABP inhibits both endogenous but also express CAT1 channels in RBL cells. And one point which is not clear so far, the amino terminus of CAT1 is able to block crack channels, endogenous crack channels, and also it inhibits in turn mast cell degranulation. I have to say, where this effect comes from, it must be a sort of unspecific effect in terms that it's not directly related to an interference with the channel formation. But we have to clearly say we never understood where this effect comes from, although it was nicely reproducible. It's a very strong effect, but it somehow is due to a certain lack of specificity. And that was the most important point I want to convey here in this lecture is do as much controls, use as many as specific tools to come to a conclusion for your actually proposed work. Okay, that's on the involvement of CAT1 or TRPV6 as a tool or as a protein behind the crack channel machinery. So that now historically, 2005 and 6, and I will now switch to the next lecture going on, where actually I want to show you the two proteins which are actually behind the crack channel machinery, and this is STIM1 and OR1. And actually, after that paper, 2000 was the Nature paper. Our paper was published 2002, which clearly says, okay, TRPV6 or CAT1 is not a subunit, is not behind the crack channel. These proteins, they have been identified by other groups, STIM1 2005 and OR1 2006. And let me just now present you actually, because it's very interesting how new proteins can be identified, because that is also can be taken as a general approach. Just going back to this general mechanism, as we just have heard, we are now looking and it's somehow interesting why it's proposed that there are two proteins involved. We have heard we get a drop in the calcium concentration due to an increase of IP3, which leads to depletion of ER calcium stores. And this drop in the calcium concentration, this needs to be signaled now to the crack channel in the plasma membrane. So we somehow need a protein which senses the drop in the calcium concentration in the lumen of the ER, and at the same time generates a signal that opens crack channel in the plasma membrane. Let me just come to models that have been proposed before actually STIM1 and OI have been identified. Of course, in all of these models, we have the crack channel in the plasma membrane. There is only a different hypothesis here in these three models, how these crack channels in the plasma membrane actually get activated. The first use you see here, because we already talked about the IP3 receptor, which releases calcium by binding IP3 and leads to a drop in the calcium concentration. At the beginning, there have been models that propose that maybe the IP3 receptor, because that gets activated, somehow that is component, the protein, which in turn interacts or communicates with the crack channel in the plasma membrane and opens the channel, so you have a sustained calcium influx. Let me say, this is a sort of conformational coupling, because we already heard it maybe in terms of calcium channels, where calcium channels show a conformational coupling with ryanodine receptors in the sarcoplasmatic reticulum. Maybe you remember the muscle contraction in smooth muscle, in hard muscle, but also in skeletal muscle, that somehow involves an interaction of a protein in the sarcoplasmatic reticulum, the ryanodine receptor, which is also a calcium channel, in a way as the IP3 receptor is, in terms of the conformational coupling between these two proteins. That was the first hypothesis. The second is, we have vesicles. And as we just have heard, that calcium that is released then via stores is important to drive the fusion of vesicles, which contain histamine with a plasma membrane. It can also at the same time help before in a way that we have vesicles which in the membrane of a vesicle have this quark channel inserted. And when we have now at the beginning of this process, just starting a release of calcium, this release of calcium that can drive the fusion of these vesicles with the plasma membrane and lead to an insertion of quark channels in the plasma membrane, which has been called the secretion model. But it turned out finally that the calcium which is released from ER, that it's not enough, that is not sufficiently high to drive this fusion process, because we've heard it already. Also for the release or the fusion of histamine containing vesicles, you need this sustained long calcium entry. But that was a second, called the secretion model. And interesting, there was also one Nature paper, but only by one group. No other groups could prove it. And meanwhile, this model is completely out of discussion, that due to the drop in the calcium concentration, some factor, calcium influx factor, it has been called, CIF, is generated by the ER, which activates a calcium channel, the quark channel in the plasma membrane. This CIF, it was never fully characterized, only by some inhibitors, but it could never be purified, and no other group than only one group thought this would be a correct mechanism, but it was not confirmed by any other group. So this is out of discussion. This calcium influx factor, let me say, seems to be not existent. What finally will be the correct model, although the proteins here, the IP3 receptor was not correct, is the conformational coupling model, in terms that the communication between a protein in the endoplasmatic reticulum and the quark channel in the plasma membrane occurs due to a conformational coupling, due to an interaction with each other. So let me just get you back, but that is for clear. That was also why we have a certain focus here on calcium signals. You can see here why calcium is so important, contraction, secretion, of instance, of cytokines or histamines, but also, and that particularly related to T-cell activation, which we will also will hear something about, that the calcium influx can also lead to an activation of transcription factors, like, for instance, NFAT, and that makes a downstream signal in the nucleus. And of course, we have phosphorylation processes driven by calcium-dependent kinases, calcium module independent, for instance. On long term, it's very clear that calcium is linked to cell growth, to proliferation, to differentiation, and this gene expression that also is a long-term process. So it's very important to understand how these calcium signal actually is regulated. T cells and mast cells, that are the most prominent ones. And 30 years ago here, in T cells as well as in mast cells, particularly in cell lines, these quark channels have been initially characterized. Later on, it was also characterized in other cells, but these were the first two initial cells. Properties we already heard, we already have seen the biophysical properties. It's highly calcium selective, it's non-voltage gated, it shows this inwardly rectifying current voltage relationship, is strictly regulated by store depletion. It only gets active when you deplete the ER stores. And of course, this protein needs to have numerous, at least 2 transmembrane regions to work as a functional ion channel. What's behind the molecule, how it actually is activated, and of course, a 3-dimensional structure in terms of a crystalline or cryo-M structure, that is not known. At the time we are now going to, it's between 2002 and 2005. So It has been proposed also from others that maybe TRIP families might be behind, but it finally turned out that was the Nature paper I just mentioned. That is actually a review, but before it turned out that TRIP V6 or CAT1 is not the protein behind. So how actually did the search for, and here's the search now for the calcium sensor in the ear, how did it continue? And there are actually two groups working in competition, but you see at the same time. So this is a very, very competitive field, and it's still competitive, although meanwhile 15 or 16 years have gone. First of all, two different cells, Drosophila S2 that are insect cells derived from Drosophila, and HeLa that are actually cells from an uterus. And the name of the women who unfortunately died of uterus cancer, that finally leads to this cell development because it's a cell line from an uterus cancer cell. And I only know the forename, that is Helen, the surname, and La and something is the family name. But these two cells, they have endogenous crack currents. So they have the machinery in terms of store depletion leads to activation of calcium influx. So how actually was there a search going on in these cells to identify components behind these crack channels? And this group, and you can see here what they actually used here, both groups, they used siRNA. siRNA against proteins where actually there is only some function known, but not clear for many. They checked 170 genes where they had siRNA against, and this group, they checked 2,304 human genes where actually they had siRNA. And this is a competition also in terms of time, who will make the race, because there is a lot to do. And of course, you cannot do these experiments usually by doing electrophysiology, because then you have a lot of cells to patch, cells actually doing controls and cells that have been treated by these specific siRNAs. But okay, that will not work. So whenever you have something to approach with many, many test samples, you need to study for an effect siRNA in terms of suppression of this crack channel machinery, you have to go to fluorescence methods. And classically here, as I've mentioned already before, there's a very nice tool that you can use fluorescence dyes as reporters of cytosolic calcium concentration. And the main point, of course, is the increase in the cytosolic calcium concentration that occurs in these cells due to activation of crack channels that needs somehow to be disturbed by siRNAs. And then, of course, we can further look on how this might be happen. So actually, there was a protein that has been known before, and the protein is called STIM, stromal interaction molecule. And you can see here, they checked at the level of RNA of STIM. So here it's not, because at that time there were no proteins, no antibodies available against STIM protein. But you can see whenever they treat a cell, this siRNA, that is a double-stranded, this is a little bit more stable than a single-stranded RNA. But in terms it functions similarly, it decreases the protein concentration, or also at the level of RNA, because it destabilizes the RNA of a protein of interest against these siRNA is made. And you can see here, when you check in cells which have been treated with double-stranded RNA against STIM, you can see here from control, there's a clear decrease. It's not fully abolished, but I would say there is a decrease, let me say, to about 30%. That you have always to have a control RNA of another protein, that is control gene presenilin, and you can see, okay, there is no unspecific harm to the cells. And now let's go to these calcium experiments. So here, in terms of fluorescence, that already calculated into an intracellular calcium concentration, which would be expected in the low micromolar to micromolar range. And now we have to understand what usually the protocol is. So I want to make it pretty clear that you understand, because these measurements typically look all the same. Here's the time scale. You see here seconds. One experiment lasts here 1,000 seconds. So that's quite on a slow time scale. But these calcium signals, they actually occur on a very slow time scale. So usually what we do at the beginning, so these are signals from single cells analysis, and each of the trays belongs to a single cell where you actually check for calcium cytoplasmic concentration. What's going on when you make these different manipulations at the extracellular side? You start with a 2 millimolar calcium concentration. Then you switch to 0, and then to 2 millimolar again. That is just to check that the cell membrane is somewhat tight against a 2 millimolar, which is a physiologically range calcium concentration. Because these channels or the crack channels should not be activated in these cells. But now you go back to 0 calcium, and at the same time you apply TG, is an abbreviation which stands for tapsigagin. Tapsigagin. Tapsigagin, it works in a similar way as EGTA, but EGTA that grabs the calcium and prevents uptake in the ER. So due to the leak, it helps to deplete the stores. Tapsigagin, it works directly on the calcium ATPase that pumps calcium back in the ER stores. That's very specific, only inhibiting the calcium ATPase in the ER stores. And by preventing calcium pumped back, the leak leads more and more over time. So that takes some time, some minutes. The leak over a certain minutes will lead to a complete depletion of the ER calcium stores. And you can see here what happening here. When tapsigagin is applied, usually you see an increase in the calcium concentration because now the leak is working. The ER leaks out calcium. The question is, why does it actually go back to this level just before application of tapsigagin? Because we have another calcium ATPase, and this calcium ATPase is sitting in the plasma membrane. This is not inhibited. That was a very, very effective tool and specific tool. This is not inhibited by tapsigagin. So this is working, and whenever calcium rises in the cytoplasm, the calcium ATPase in the plasma membrane pumps calcium out, but there is never a calcium pump back into the lumen of ER, because here in the membrane of the ER, this calcium ATPase is specifically inhibited by tapsigagin. So that is here actually at that time point. The stores are completely empty of calcium. There is no calcium in the stores anymore. So based on the mechanism, the calcium channel in the plasma membrane, they need already to be fully activated at that time point. But because there is no calcium in the extracellular medium until that time point here, we do not see an increase in the cytoplasmic calcium concentration because there is no calcium that can mediate it. But when we switch now here again to a 2 millimolar calcium containing solution, you can see here this very nice increase, and this all occurs because we have an influx of an influx of extracellular calcium into the cytosol. So the idea here is also by using a store depletion in the absence of extracellular calcium. So whatever we see here only occurs due to a calcium out of the stores. But whenever the stores are depleted, then there is no calcium in the cell anymore. Whenever we switch to 2 millimolar calcium at the extracellular, the outside, whatever we do see here is the influx of calcium through the crack channels that are already, and that is the reason why it increases here so fast, because the stores at that time point are already completely depleted. So we have fully activated crack channels here. When you now see what happens when these RNA or siRNA against STIM leads to a knockdown of STIM proteins, you can see here everything looks pretty the same. Here you see the release of calcium, but whenever we go to a calcium-containing extracellular solution, what's completely missing here is the influx of calcium. So it seems that this siRNA highly efficient, inhibits the influx of calcium over these calcium channels. The question is, where does it actually act? And that is another test in these JKU T cells that are now the T lymphocytes. They also have this sort of crack current, as I mentioned before. These are signals of single cells, wild type, so untreated or control treated. These are now with siRNA treated against STIM. And these are, let's just directly focus on the average signals here. These are only typically the average, which you usually take. You can see here almost the same, 2 calcium, 0 plus tapsigargin. So this is already depletion of the stores. And then they do a twofold switch in the extracellular calcium concentration to 0.4 millimolar, and this is to 2 millimolar. So maybe we directly look at that signal here because that relates to the experiment we've seen before. You can see here, release quite undisturbed when STIM1 is knocked out. However, with 2 millimolar calcium, these cells, it's not completely inhibited, but you can see here from that signal here, there is still a substantially significant decrease in the calcium influx when STIM1 protein is knocked out. So that is the first characterization you typically do. You can imagine when you have to do such experiments with 2 or 3,000 siRNAs, that only can do automatically. And what you typically are used here are plate readers. Plate readers where you have these 96 well plates or even larger 384 plates, where in each well you have those cells, and in each well maybe you put in a different siRNA so you can check at the same time, just in terms of a high throughput testing, so that you very within a certain time frame come to results. Which of those siRNAs lead to an inhibition with these crack channel currents, crack channel influxes? Of course, you can do when you have already established one or those, and here particularly the siRNA against STIM, that this works highly efficient. You can also do these single channel, not single channel, single cell, whole cell recordings. You can see here in Drosophila cells how this current actually develops. That is what we've seen before. That is current, calcium current activating over time. It takes some time for the stores to get depleted. Same protocol as before. That is the calcium current activated with this nice current voltage relationship with this highly positive reversal potential. And in this cell, when this cell is treated with siRNA against STIM to knock out STIM protein, so this was a fully efficient effect. You can see there is no current activating anymore. So the question is, how does, and that's of course, it's known, it's not the structure by itself, but at least from secondary structure prediction that are bioinformatic analysis, you can already get some information how this protein, this STIM protein actually is composed. Which domains can we find in the STIM protein? The first of all, it is a single membrane spanning sequence. Let me just start in the middle, because that was of course expected. It should somehow sit in the ER membrane. And of course, one part of the protein needs to be localized in the lumen of ER, because as we've heard, this might be a protein which senses the drop in the calcium concentration first, because When you have the stores depleted, then a signal is generated to activate the cracked channels in the plasma membrane. You can nicely see here, this is in the lumen of the ER. And here are certain domains, SAM domains. These are protein-protein interacting domains. But one domain we already know for sure. This is an EF-hand domain, and that fits very nice to our hypothesis. EF-hand domain is a calcium binding domain. So this part at the lumen, in the lumen of ER, this could be the sensor for calcium. And by sensing a drop in the calcium concentration, meaning when calcium dissociates from that domain due to the drop, it might be a signal generated that is conveyed to the cytoplasmic side of this protein. And you can see here, when we have a closer look at the cytoplasmic side, you may remember these regions we already have heard, cold regions. These actually are very important regions or domains that are known also to be important for protein-protein interactions. So they allow the communications between proteins, because one cold domain can very nicely interact with another cold domain, and by binding to each other, there can be, for instance, in our case, the cracked channel might be, for instance, activated. We have heard about these domains, cold regions, when you remember in the process of vesicle fusion in the synapses, but that was in biological signaling 1. There are snare proteins that actually bring these vesicles closer to the presynaptic terminal membrane, and the snare motif actually is composed of cold motifs. So that nicely fits. We have here, it sits in the ER membrane. It can sense the calcium concentration here in the lumen of ER, and it has protein-protein interacting domains at the cytosolic side. That potentially might communicate with the calcium channel in the plasma membrane. So what can be further done somehow to make this story more conclusive? What you can see here is whenever wild-type STIM was overexpressed, and you can see it here in cells, this is a blank transfected, so this is a control. Keep in mind, whenever you do a transfection of cells, maybe you have learned it in our cell culture practical course, whenever you do a transfection, it's a treatment of the cells. Of course, here we overexpress STIM1, but keep in mind the correct control would also be to treat the cells with the same reagent you use to overexpress STIM1. That means to get STIM1 plasmids into the cell, but you do not use STIM1 here. Maybe only typically STIM1 is tagged here by fluorescent proteins, so you can identify those cells that actually lead to an expressed STIM1 protein. Here you can also use the dye and overexpress the dye in the cells, but that's the correct control, because also treatment by these reagents that allow plasmids to get into the cells, for instance, lipofectamine or transfectin, there were a lot around, they could also lead to a change in the behavior. And you have to do the correct control here. But you can see there's not much change. Maybe here with a lower calcium concentration, the signal is a little bit higher when STIM1 is overexpressed. But what have been done here, the EF hand is the domain that binds calcium as long as the stores are full. But when the stores are now depleted, there is no calcium sitting anymore in the EF hand. So can we somehow simulate that there is no calcium at the EF hand side? And that's can be done because the EF hand, it contains negatively charged amino acids for binding of calcium. And it is well known that one or two of these negatively charged amino acids, they only need to be neutralized, for instance, by alanines, that you can switch from a structure in EF hand that can bind calcium to a structure that is not able to bind calcium anymore. Meaning there has been an EF hand mutant of STIM generated where now the efferent has been affected in a way that it cannot bind calcium anymore. So there is no calcium sitting, indicating or leading to a state that looks like the stores are already depleted, although the stores are full. But now the efferent has disturbed in a way that it cannot bind calcium anymore, although the stores are full. And let me now give you here the point where you have to look here at that signal here. Because you can see here, that is the beginning with 2 millimolar calcium, where typically the calcium, the crack channel should be closed. It should be closed because the stores are not depleted. But what you can see here is, interestingly, without store depletion, the stores are full. We already have here a high increase in the cytoplasmic calcium concentration. And you can see here, we can do it. Calcium 2 millimolar, 0, 2 millimolar calcium. Afterwards, here, tapsigargin is applied. And what you can nicely see here with 2 millimolar calcium, here it's already an increased cytoplasmic calcium concentration. Here, again to 2 molar, it even gets higher. And afterward, this looks pretty much the same. But the most important point is, at the very beginning, despite the fact that the stores are full, we already have here an elevated calcium concentration. And this could be because STIM1, the efferent mutant, is already in an activated form, meaning it looks like, or it is in a state as there is a calcium depletion has already occurred, meaning STIM1 is activated, and by this, it activates the calcium channel of the plasma membrane. So here you can see that are these mutants actually that have been used, the efferent mutants. And this increase in the cytoplasmic calcium concentration with the efferent mutant, that in turn has been checked whether this increase shows already the characteristics by certain inhibitors that have been known to inhibit crack channel calcium influx. And we already have heard about 2-BB, and you can see here the calcium signal looks the following way here. We have here now the efferent mutant. It's already in an activated form because there's no calcium bound anymore. So this is the calcium influx from 2 millimolar to 0, from 2 millimolar. Sometimes this is higher because whenever the cells has calcium channels open all the time because they are pre-activated due to this activated form of STIM, the cells somehow find some counter-regulatory mechanisms that the calcium doesn't get too high. But when you go from the very beginning with 2 millimolar to 0, okay, there's a drop. And then when you go to 2 millimolar again, maybe this increases to a higher extent. When 2-BB is applied, you can see here, very nicely inhibited. Another inhibitor at that time was SK and F. And you can see here 2 millimolar calcium applied for a second time when SKF is given, not completely inhibited, but also nice inhibition. And not a very specific anorganic ion, gadolinium, but it's highly used for inhibiting, for inhibiting calcium channels, similar as lanthan, also with free positive charges. And you can also see here, inhibited. So it means that the basic characteristics, the characteristic in terms of inhibitory profiles by these three compounds that have been known to inhibit crack channel influx, this elevated calcium concentration we have seen here seems really to be related to an increase to an activated crack channel that leads to an increased calcium influx. So that's very nice because it shows that it's in a way somehow STIM1 seems to activate the crack channel or is involved in activating the crack channel. So let me just give you a further clue on how fluorescence microscopy can be used here in terms of labeling of STIM. You can see here, this is 0 minute, a cell where STIM is expressed, labeled at the luminal site with an YFP. And here you see a CFP-ER marker. So you have, against each organelle, you have markers that can you use somehow to get an idea where actually is the STIM1 expressed. When it is expressed in the membrane of the ER, then the CFP-ER marker, they both should, because the CFP and YFP, the expression pattern should be quite similar because both are sitting in the ER, and you can see it here. Usually, although it's yellow and blue, usually it's used a false color code. Green and red, because when you do an overlap now of these two images from the same part of the cell, wherever you have two of these proteins expressed, meaning you have a color of green and red, it will end up with a yellow mixture color. And you can see here the expression pattern of the ER marker nicely correlates with expression of STIM1. So that was a further clue that indeed STIM1 is sitting in the ER membrane. So what happens when you treat the cells with tapsigagin? And it's a very, very interesting and crucial experiment because tapsigagin depletes the stores. So do we see something change in the distribution of STIM1 when the stores, the ER stores, gets emptied of calcium, meaning calcium dissociates from the EF hand? And you can see here what is happening. Here, that is the ER marker. So there is not much change, but have a look how STIM1 redistributes. What you can see here of course, it's not a complete uniform distribution, but here now you see these clusters form. You get these punctate structures of STIM1, particularly also close to the plasma membrane, but also inside of the cell. So it seems something is going on when you deplete the ER stores of calcium. And that, of course, that's a further indicator that STIM1 is reacting in terms of as a calcium sensor when you deplete the stores. When you have a look at the mutant, you see here a D is substituted by an alanine, a negatively charged aspartate by an alanine. This mutant cannot bind calcium anymore. So even at 0 minutes, when the stores are full, this mutant should already have a pattern that is more similar to that pattern of wild-type STIM when the stores are depleted, because there is no calcium bound to the mutant. And you can see, although it doesn't look the same, but you already see here these clusters, these punctae formed. At least it looks different to the wild-type distribution of STIM. And you can see here when this STIM1, which already seems to be in the activated form, when now the stores are depleted, you can see by tapsigagin, there is not much change going on. That was expected because this STIM1 is not able to bind calcium anymore. It will not react to a change in the calcium concentration from a high calcium containing ER when it's switched by tapsigagin to a depleted ER. So that at least is in line that the EF hand is the prominent domain within STIM that senses the calcium concentration. And by dissociation of calcium from the EF band, you can see here this redistribution in terms of these clusters or punctae-like structures of STIM1 are formed. So the question, of course, was, is STIM1, what is it doing? Could it be the calcium channel, or how is it related to the calcium channel? But keep in mind, it's only a single transmembrane protein. So there are so far no single transmembrane channel is around. So the following models, or the following two models, have been developed. We have STIM1 particularly sitting in the ER membrane. And you can see here the blue or the purple dots, that is the calcium in the ER. When the stores get depleted, you can see here then these clusters, these punctae-like structures of STIM1 are formed. And now there are two possibilities. Here is the calcium channel, the crack channel in the plasma membrane, and it's waiting to get activated. One could be that the cluster is inserted in the plasma membrane, and by an interaction with the channel, it activates the channel and allows a calcium influx. The other possibility is that we have sites where the ER membrane gets close to the plasma membrane. Those sites, actually, they have been known that they exist, that are plasma membrane ER junctions, because they are not for the whole length of the plasma membrane, but at certain spots here. We have these ER plasma membrane junctions, and the idea here is at those sites, STIM1 in an activated form, when the stores are depleted, can reach onto the plasma membrane, find the crack channel here, and activate the crack channel. So at that time, 2006, it was not clear how it actually worked, but meanwhile, it's clear this model is the correct one. So let me just summarize at the level of STIM. So what are the characteristics of STIM? It has an N-terminal EF hand motif. That is the calcium binding motif, which functions as a sensor motif and senses the drop in the calcium concentration when the stores get depleted. This is a single transmembrane, a single ER transmembrane protein. It has at the cytoplasmic site, also called cold domains, that could work as protein-protein interactions domains. It localizes in the ER, but it could also go to the plasma membrane. At that time, people believed that the punctate structures, actually, these punctae are in the plasma membrane. But finally, it turned out these punctae are formed in the ER, but they are in these ER plasma membrane junctions. So they are very close to the plasma membrane. The fluorescence microscopy, they do not have a high resolution enough based on the criteria by light, by excitation, that you can differentiate whether it still sits in VR, which is very close to the plasma membrane, or it has already moved to the plasma membrane. But meanwhile, it's known that the higher part of STIM1 keeps localized to the ER membrane. So STIM1 plays a dual role. It senses the luminal constant store, and that's not true. It don't traffic to the plasma membrane, but it traffics to the ER plasma membrane junctions, where it forms these punctae and activates the crack channel. So as a final conclusion, as I just mentioned, because it's a single membrane spanning region, it's highly, no increase in calcium upon STIM1 overexpression. That is also we have seen. If this would be the channel, there would be expected really a dramatic increase. But particularly single membrane, it is highly unlikely that STIM1 by itself forms the crack channel. And here I will stop now, because next time we will have a closer look how actually the calcium channel in the plasma membrane has been identified next 1 year later in 2006. Okay, that's for today. We see again next Thursday. So have a nice week.
Thank
Bye.
you. Bye.
Thank you. Bye.
Thank you. Bye.
Thank you. Bye.